Oh, I hope that works. We'll see. Okay, let's go ahead and get started. Sometimes technology is not my friend. Okay, that'll work. RNA stands for ribonucleic acid. Oh my God. I'm looking to move you. Okay, so RNA stands for ribonucleic acid. This actually comes just like deoxyribonucleic acid. This comes from the sugar that is found in the molecule of RNA. Okay, so type of biological molecule, I'll just kind of move this back and forth. This is a nucleic acid. There's two types of nucleic acids, DNA and RNA. Can we see that okay? Is that good? Okay. We talked about, in chapter 2, 4 different biological molecules. We talked about proteins, carbohydrates, nucleic acids, which we're talking about more now, and lipids as well. Okay? So RNA is one type of nucleic acid. Its structure is it's got some similarities to DNA and some differences. Okay? One major, this guy right here, this represents our DNA or RNA. So label this RNA. And then this guy, of course, right here, this guy is DNA. Okay. The first major difference between the two structure-wise is this guy is a single strand, still twisted, and this guy is a double strand. So under your structure, the first thing we are going to write is we're going to write single-stranded. So one single strand. Our RNA molecule is made up or composed of nucleotides. And if you guys remember, nucleotides are made up of a sugar, a nitrogen base, and a phosphate molecule. So RNA is composed of nucleotides, and those nucleotides have a sugar molecule, a nitrogen base, and a phosphate. Oh shit, I didn't mean to do that. Stupid technology. Okay, so let's look at, I'm going to put NB for nitrogen base. I think you guys can remember what NB means. There's 4 different nitrogen bases in RNA. There is adenine, which you guys are familiar with. There is cytosine, which you're familiar with. There is guanine. There's no thymine. But we have a different base that starts with a U. So U stands for uracil. Okay? So we still have the same base pairing rules. Okay? So let's put base pair rule. Extremely similar. Okay? So we have A is going to match with U. Okay, that's one scenario. C matches with G. We'll just stick with that. Okay. So no T, but we've got U instead. And then the last one, and I am running out of room, is our sugar. Our sugar is not deoxyribose, it is called ribose. That's a little bit better. Okay, so if you come over here to your picture, These represent our nitrogen bases. And if you look, these two are identical, these two are identical, these two are identical. This is where there's a little bit of a change. You've got this CH2 here, but you don't have one here, okay? So the bases found in RNA, this is cytosine, this is guanine, this is adenine, and this is uracil. And then in DNA, cytosine, guanine, adenine, and thymine. So the big difference is that nitrogen base. Now there's 3 types of RNA that we have to be familiar with. I'm wondering if I need to switch pins to get this all in. Okay, so the first type, number 1, is called mRNA, and that is messenger RNA. So mRNA stands for messenger RNA. His function is to make a copy of DNA and take the information out of the cell. Okay? So make copy of DNA and then take out of nucleus. I said cell. Take out of nucleus. So DNA cannot leave the nucleus unless we're dealing with cell division. And if you guys remember, in prophase 1, the nucleus disintegrates, it disappears, the chromosomes align themselves in the center of the cell. You guys remember all that? Chromosomes are pulled apart, cytokinesis happens, all that good stuff. So that's the only time that chromosomes or the DNA can leave the cell. Okay, it's basically stuck inside of the nucleus. And the only way to get the information out is to have another molecule make a copy of it and then take it out of the nucleus. So that's the job of messenger RNA. Okay, your second type of RNA is called tRNA, and that is transfer RNA. And his function, well, he kind of hangs out in the cytoplasm. His job is to hang out in the cytoplasm. He goes around, he snags amino acids, and then he brings them into the ribosome so proteins can be made. Okay? So his function is to grab amino acids and bring into ribosome to make proteins. You guys see that okay? Still good? Okay, you have young eyes. Okay, and then our very last RNA is called rRNA, and that is ribosomal. And ribosomal RNA, its main function is to help with the structure and help in making ribosomes. Okay, so function, help to make ribosomes. Ribosomes are important because that's where proteins are actually made. Now, as we move on to protein synthesis and the first step of it, we are going to pay close attention to the first 2 types of RNA. Those are the ones that play a really big part. Okay, so move to your protein synthesis. It's a little bit better, kind of. There we go. Okay, I'll just kind of move it around, I guess. Okay, so we're going to have to start by writing some information in the margin. So up here, I am just going to write the definition or what protein synthesis is. So this is process by which proteins are made. So the term synthesis means to make. So if it's protein synthesis, we are making proteins. Okay? I'm going to start over here on this side. Where does protein synthesis happen? It happens in 3 places in the cell. Okay? This is our cell. I'm going to draw myself some DNA in here. Just like that. So the first place that we've got any sort of action going on is in the nucleus. So we've got a lot of action going on in the nucleus. The ribosome is the second place. We've got a lot of stuff going on in where the magic happens, in the ribosome. And there's also a little bit of stuff happening in the cytoplasm. So if it's a eukaryotic cell, we're going to have protein synthesis happening, nucleus, ribosome, cytoplasm. If it's a prokaryotic cell, like a bacteria cell, there's no nucleus. So the only place that proteins are going to be made are in the cytoplasm and in the ribosome. Okay? Okay, so I'm going to move over here and I want to look at what are proteins. Now you guys already know this because we've learned it in chapter 2, but I will remind you. They're biological molecules composed of amino acids. Now, if you remember, biological molecules are long molecules that are composed of smaller molecules. Okay? And the smaller molecules are amino acids. All of the biological molecules you learned about, lipids, are made up of 2 smaller molecules called fatty acids and glycerol. Carbohydrates are made up of simple sugars or monosaccharides. Does this kind of sound familiar? Proteins are made up of amino acids. Okay? And then nucleic acids are made up of nucleotides. So when we're talking about proteins, the small molecules that make them up are amino acids. So why are they important? We're going to label that as function. They have lots of functions. Okay? One major function is to function as an enzyme. So enzymes speed up chemical reactions, or they even they lower the activation energy to make them start or go faster. There's lots of chemical reactions happening in our cells, and they have to happen quickly, okay, in order for stuff to happen. Okay, so they function as enzymes. They function for gene expression. And this is what we're dealing with right now is gene expression. That means that in living organisms, in order to see the traits or the phenotypes, the genes have to make proteins, and then those proteins exhibit the traits, okay? So like Natalie has got very curly hair back there. She's got naturally curly hair. So she has the gene for curly hair, okay? So that means her cells produce the proteins to make curly hair. Olivia, on the other hand, I don't think has the gene for curly hair. So that means she's producing the genes, okay, her DNA has the genes for straight hair. So they're producing proteins that give you straight hair, okay? So proteins in your cells exhibit the different traits that we have. So that's what we mean by gene expression. A few other things, they function as hormones. so I'm sure you guys have heard of like estrogen, progesterone, that's some testosterone. Cholesterol is actually a hormone as well. So hormones, the last one is cell structure. So a lot of times proteins are going to be found in like the structure of organelles, making up our different organelles. Okay, so central dogma of molecular biology, that is a super fancy way. When we're talking about central dogma, we're talking about like the major processes in living things that make, the easiest way to say this, all of the most important processes happening in living things. And it has directly to go with genetics and passing on your genes and exhibiting different proteins. It always kind of happens in a linear process. And there's three major processes that make up the central dogma. So 3 processes. You guys know the first process. It's DNA replication. So these are things that happen like in all living things. This is the central dogma. Okay? So DNA replication. The second one is called transcription. And that is actually the first step in making proteins. And we'll talk about that in depth in just a second. The third process is called translation. And that's the second step. So over here, what we've done is they've just kind of illustrated the central dogma, not DNA replication. So this step from here to here, this is called transcription. And the bottom line, transcription is having RNA make a copy of DNA. So mRNA makes a copy of DNA. And this is step 1 of making a protein. This guy is step 2 of making a protein. This guy is translation, which we'll talk about tomorrow. And if we're just to sum this up, this is mRNA making proteins. mRNA making proteins. Okay, so that's kind of protein synthesis in a nutshell. We are going to flip this over and we're going to look at transcription today and translation tomorrow. So this is where we are going to use your colored pencils. Okay, I am going to start up here in the margin. And I'm going to make an arrow and we're going to say process by which mRNA makes a copy of DNA. Okay. I need to unzoom this so you can see the big picture. Okay, so it's a little bit better. So all of this right in here, okay, this is all inside of the cell. This is inside of the nucleus, excuse me. This is our nuclear boundary, okay? So right here, this, and this is representing the nuclear envelope. Okay, so there's not a special spot for it, but we're going to write it in envelope. Okay, now there are little spaces in the nuclear envelope or the nuclear membrane, And they're literally little holes, and they're called nuclear pores. And that's how things go into the nucleus and how things leave the nucleus. So this right here and this right here is representing the nuclear pore. That's where, like I said, stuff goes in and goes out. So this is inside of the nucleus. So we're just going to make a note of that, inside nucleus. And of course, out here, we're going to label it right here. This is going to be in the cytoplasm. So inside of the nucleus, outside of the nucleus. In this spot up here, we are going to go ahead and write DNA to mRNA. That's our goal, is to take DNA and make a copy of it with RNA. We just talked about this. What are 3 different ways RNA and DNA are different from each other? What's the very first major thing that you think of? How is DNA and RNA different from one another? Good, single strand. What's the other thing? One more that's perfect. Your cell? Not. Your cell, not thiamine. Very good. We got one other big difference. Louder?
There's only one ribose instead of 2.
Well, it's ribose and not deoxyribose. Okay, so correct. Okay. So let's break this down into steps. So we're going to say step number 1, very similar to replication. And we're going to say DNA helicase unzips molecule to separate strands. So I'm going to now take my colored pencils and I'm going to start with blue. And I am going to label this guy DNA. That's kind of hard to see. DNA. And I am going to color the backbone of my DNA molecule. I'm going to color blue. You choose what color you want. Because I want to be able to tell what's DNA and what's RNA. So our DNA molecule has unzipped and it is separated just in one specific spot. When RNA is making a copy of DNA, it does it per gene, so it's just a chunk. It doesn't happen to the entire DNA molecule. That messenger RNA isn't made from the entire molecule, but in very kind of smaller chunks. So all of the blue is DNA. I think I'm just going to lightly color this in. DNA. Now, down here, I'm going to label this as my original strand. So, original. So, this really is a semi-conservative process as well because we're using an old strand to make a new strand. In this case, it's a new strand of messenger RNA, not DNA. Okay? Okay, so step number 2 up here. We're bringing into play a new enzyme. It's called RNA polymerase. RNA polymerase. His job is to bring in new mRNA nucleotides to make a copy of DNA. So the thing is we are going to make a copy of DNA into RNA. So I am going to use green for my RNA polymerase. So right here, down here, I'm going to label this RNA polymerase. I'm going to make him green. And this structure right here represents my polymerase. So it's like he's going to go through and he is going to make a copy. Okay, so he's going to bring in nucleotides to make this new strand, which is mRNA. Okay, so we're going to label this guy mRNA, and I'm going to color my messenger RNA red so I know which strands are DNA and which is RNA. So this is my backbone. And this is RNA, messenger RNA. So this DNA polymerase constructs this new strand of messenger RNA. Okay? He makes this strand using the information in DNA. So he's making a copy of it. So T is going to match with A, so we're going to fill in the bases on the RNA. Let's see if I can make that bigger. There we go. So A, we don't have any thymine in our RNA up here, but we have uracil. So A is going to match with U. C is going to match with G. G is going to match with C. C is going to match with G. T is going to match with A. G with C. G with C. We don't have thiamine, but we have uracil, so A goes with U. T goes with A. C goes with G. And G goes with C. This guy right here, see how we have 3 nucleotides, or I should say 3 nitrogen bases, 3 nitrogen bases, 3 nitrogen bases. When you're looking at 3 nitrogen bases at a time on a DNA molecule, we call it a triplet. So it's like the messenger RNA comes in and makes the DNA, let's start over. RNA polymerase comes in with the messenger RNA in groups of 3, and so it's following every triplet on your DNA molecule. So that is considered a triplet in DNA. It's called something different in RNA, but okay. So I got 2 more things to write. So our last step, step number 3, messenger RNA is going to leave the nucleus, move into cytoplasm, and then go to the ribosome. His goal is to get himself to the ribosome. So the last thing you're going to do is right over here. This messenger RNA is leaving out of the nuclear pore, and he is moving to the ribosome. And that's where all the magic happens. That's where those proteins are actually made. and that is transcription. So protein synthesis has two steps, transcription and translation. We just covered transcription, and that's just basically making a copy of DNA into messenger RNA. Messenger RNA leaves the nucleus and is going to go to the ribosome, and then we'll hit step number 2, which is translation. Okay? I'm going to give you a second to, if you need to take your colored pencils, put those away, and grab yourself a textbook. We don't have too much time, but you got about 5 minutes that we can start, and we'll finish it tomorrow. So take a second.